All right, so this video is going to run through these inheritance practice problems. So we're going to run through some Punnett squares here. We've got all of our different definitions for our various terms dealing with inheritance here at the top of each of these sheets. This will be uploaded on Canvas for you, as well as the inheritance practice problems. So I suggest that you open that up as we go through this practice so you can see what problems I'm actually working. Okay, so let's walk through these definitions first. So we've got our phenotype is our physical expression of a gene. So this is what we actually see. These are the proteins that are actually being made. We've got alleles, which are just different versions of a gene. So you could have an allele for a widow's peak or allele for the absence of a widow's peak. You could have an allele for brown eyes, an allele for blue eyes. Those are the same genes, but they code for different versions of that gene. Okay? Then we get into genotype. These are the actual alleles that someone has. So we have some alleles that are dominant that cover up recessive alleles. We have some that are recessive that get covered up. So in order to have the recessive phenotype, your genotype has to be homozygous, which means that both of those alleles are the same. So we've got two recessive alleles in order for it to be expressed. Otherwise, if you're homozygous dominant or heterozygous, you have the dominant phenotype. Okay, so the genotype, we're looking at what alleles they have. The first few problems, we'll look at specifically dominant recessive relationships. Okay, so within our first example here, in this inheritance practice problems, and I'm going to go ahead and put that here so we can see it. We've got green, capital G, we show our dominant allele with a capital letter, is dominant color for pods of peas. Yellow, which is the lowercase g, is recessive. What are the phenotypes for the following genotypes? So we've got big G, and so those are the same, so that's going to be our expression. So here we've got green as our phenotype. We've got big G, little g, and so here our alleles are different. This individual is heterozygous, and so we need to look at which of the alleles is dominant. In this case, the big G is green, so this phenotype is also green. Now we have a homozygous recessive individual, so both of the alleles are the same and they're the recessive allele, so this phenotype will actually be the yellow. Okay? And so now we're going to look at these next couple of problems. We've got 1, 2, 3, and 4 on this first page that we'll look at. And so we've got big G, and big G, little g. So we've got a homozygous dominant individual and a heterozygous individual. So the way that this works is we take the first parent, in this case our homozygous dominant, and we put each of the letters above the open spaces for these boxes. Then we take our second individual, our heterozygous one, and we put them on the side. And now when we do these Punnett squares, these genetic crosses, we just match up the letters. So this big G is going to come down to this box, and this big G over here is going to go right into this box. Big G is going to come down here, little g is going to go right into this space. Again, big G, and big G, little g. Okay? And so now we've completed our Punnett square, and now we can get into this other information here. Okay? So here it's asking for the genotypic ratio. So what are the ratios of the various genotypes that we have? So when we write these, the first thing is for genotypic ratios, homozygous dominant to the number of heterozygous to the number of homozygous recessive. So in this case, we have 2 homozygous dominant, 2 heterozygous, and 0 homozygous recessive. Then for phenotypic ratio, we're looking at the number of green pods to the number of yellow pods. So in this case, all of these, because each of them has at least one dominant allele, are going to be green. So we've got a ratio of 4:0 here, because we'll have 4 green pods and 0 yellow pods. Okay? This next example, we've got 2 heterozygous individuals in a cross. So we'll have a big G here, little g in this box, big G on the left, followed by a little g. Okay, so big G comes down, big G comes to the right, big G up here comes down to this box, little g over, big G, little g, then down here, little g, okay? And so now we can look at these ratios again, okay? And so our genotypic ratio, how many homozygous dominant do we have? Just the one. How many heterozygous? We've got 1, 2. And then how many homozygous recessive? We've just got the 1. So here our genotypic ratio is 1 to 2 to 1. So we can now look at our phenotypic ratio. So how many green and then how many yellow? So if they have at least one dominant allele, they're going to be green. So we have 1, 2, 3 green, and then 1 yellow because it's homozygous recessive. Okay. So now let's take a look at number 3. We're now going to look at a different type of trait here. In a certain species of animal, black fur, capital B, is dominant over brown fur, a little b. We want to predict the genotypes and phenotypes of the offspring whose parents are both big B, little b, or have heterozygous black fur. Okay, so we can do this cross here, and we'll do it here in number 3. Both of our parents are heterozygous, and we can do our quick cross, get our big B, big B, big B, little b, big B, little b, little b, little b. And I'll tell you that typically the way that we write these is the capital letter is typically written first. If you don't put it first, it doesn't make a difference in your phenotypic ratios or your genotypes, because you know if there's one dominant allele, then it's going to cover up the recessive. So you still know the phenotype even if you put the little letter first, but you'll see that as I'm doing this, I will always put the capitalized letter first. Okay? And so we can take a look over here. Let's see, now we're jumping into percentages. So how many of these are homozygous black fur? Well, 1 is homozygous black out of a total of 4. So we have 1 in 4 here, which you could put that into a calculator. You get 0.25, which is 25%. Looking for heterozygous, we've got 1, 2 out of 4. 2 out of 4 is 50%. And then homozygous brown fur, we've got another 1 out of 4, or 25%. Okay. Now we can look at our phenotypes. Looking at black fur, again, black was our dominant allele. So we have 1, 2, 3 out of 4 are going to be black fur. 3 out of 4 is 75%. Brown fur, you've got 1 out of 4, or 25%. Okay? And so there's that example there. Number 4, widow's peak is dominant to no widow's peak in humans. Cross a homozygous dominant female and a homozygous recessive male. Okay? So we're going to use W here for widow's peak. And so we have a homozygous dominant female, so big W, and a homozygous recessive male. So we can do our cross, big W, little w, and so on. Okay, you'll see W's not the best letter in the world because big W and little w look very similar to one another, but this is what we get. Okay, and so we get this cross and we see that all of our offspring are heterozygous. Okay, so for genotype, heterozygous. For our phenotype, they're all going to have widow's peak because they all have at least one dominant allele. Okay, so I'll end this video here with just normal dominant recessive relationships. The next video will look at some incomplete dominance and codominance. Okay. All right, so let's jump back in and look at number 5 here. So in number 5, we've got a cross with incomplete dominance. You've got a red flower with a white flower. What are the results? So let's look up here at what incomplete dominance is. So we see that neither of our alleles is dominant. our phenotype for the heterozygotes is a mixture between the two alleles. Okay? So if we've got a red flower, those are going to have to be the same alleles. Okay? And so what we'll do is we'll write a big R, okay? Because we know it's homozygous in order for it to be red. Then we also have a white flower, also needs to be homozygous, but rather than using a little r, which would show that it's recessive, we're just going to write it as a WW. Okay? And so now we do our cross just like we did before, but now, interestingly, we see that they are all heterozygous, which was to be expected. But now when we look at our phenotype, we see that these are not going to be red or white. They're actually going to all be pink flowers. So that was number 5. And so we get the mixture between red and white, and they're going to make pink. So we can hop over to 6 here. We've got another incomplete dominance cross. We're going to get a pink flower with a white flower. Okay, and then we're going to look at these various percentages here. Okay, so if we've got a pink flower, as we know from over here, it needs to be RW, and a white flower was WW. And so we do our cross here. Oh, little mistake there. WW. Here's our other RW and WW. Okay? And so our question is, what percentage of the offspring will be white, red, and pink? So how many are white? So in order to be white, you have to be homozygous for that white allele, and we've got 1, 2 out of 4. So that's 50% white. Okay? How many of the offspring will be red? In order to be red, you've got to be homozygous for the red allele, like we saw here, but none of them have that. So we've got 0% red, and then we look for the percent pink, and we've got 1, 2, so we're 50% pink. And so those are our incomplete dominance examples, okay? Where when we have this heterozygous individual, where they have 2 different alleles, they're going to be a mixture between what those other 2 allele phenotypes are, okay? Number 7, we're now going to jump into codominance. Okay? Human blood type is one of the best examples of codominance that there is. And so we're going to run through this example, okay, in which with blood typing, the A allele is codominant to the B allele, and then the O allele is recessive to both. So we've got an interesting relationship here with blood typing, okay? And so codominants are where both alleles are expressed in an individual that is a heterozygote. So if they have both alleles, both are actually shown. An example that you typically see in a textbook will be a red flower and a white flower. And then rather than making a pink flower that we see in incomplete dominance, in codominance, that flower is going to be both red and white. Okay? So number 7 reads that we have a mother with AB blood type. So we've got an A allele and a B allele, and a father that is homozygous B. So that tells us that both of his alleles are the B allele. So what potential blood types can their child have? So we just do our simple cross, AB, AB, BB, and BB. So it looks like here, we're looking at our genotypes. What possible blood types can we have? That child can be AB or BB. And so those are our 2 potential blood types here. And so what we see in blood typing, and this is discussed in the lecture, is that we have antibodies or antigens of both of these on our red blood cells. So both are expressed. Okay? In 8, we've got another example of codominance. We have a mother that is heterozygous A. So in order for her to be heterozygous A, one of her alleles is an A, but then if it's heterozygous, she's got to have different alleles. If she had a B allele, she'd then be AB, so she must be AO. And then we've got a father that is heterozygous B, so he's BO. And so now again, we're looking at what potential blood types the child can have. So we do our cross, and this child could have AB blood type, they could have A blood type, B blood type, or O blood type. So this is a scenario where the child could have one of any of the 4 possible blood types that there are. Okay? So let's jump to number 9 is another example of codominance. Here we have a mother with type O blood and a father with homozygous B blood. And so we do our cross here. And any children that they have to have B-type blood. And if they don't, something very fishy is going on here. Okay? So now we're going to jump into 10, 11, and 12, which all deal with the sex chromosomes. And the sex chromosomes are special because they deal with sex. And so we have traits that exist only on the X chromosome or the Y chromosome, although most of them exist just on the X because it's a much larger chromosome. So 10, we're just going to do a typical cross between a mother who is XX and a father who is XY. And so we're looking, what are the odds a child is male? What are the odds a child is female? So we perform our cross and we see that in each scenario where a child is born, you've got a 50% chance, or 2 out of 4, that it's a female, and a 2 out of 4, or 50% chance that that child is male. Okay? And so when we're doing sex-linked traits, we always have to have these X and Y chromosomes in there because the alleles aren't inherited the same way as our non-sex chromosome alleles. Because the chromosomes themselves are different, sometimes we have a gene on one and not on the other. So number 11, we're going to deal with an X-linked trait. So it exists on the X chromosome. Females are XX, males are XY. And we're going to look at color blindness, which is a recessive allele. So if you have a mother that is a carrier for the color blindness gene, which means she's heterozygous. So we've got X, and for color blindness, I'm going to use a B. So she's got normal vision, so we know that she has at least one dominant allele. But we do know that she's a carrier, okay? And so she's got an X little b, okay? Dad's got normal vision, so he's X big B. But this is an X-linked trait. It does not exist on the Y chromosome at all, okay? So we put nothing on the Y. And then we do our cross as normal. X big B, X big B, X big B, X little b, X big B, Y, X little b, Y. So we've got a couple of things that we can look at here, and this specifically looks at what are the odds they have a child that is colorblind. And so this daughter, normal vision. This daughter here has the recessive allele, but also a dominant allele, so she's a carrier with normal vision. This son has normal vision, but this son right here has color blindness. So if we're looking at the chances of a child having color blindness, 25%. If we're looking at the chances of a son, specifically looking at a son with color blindness, if they have a son, 50% of the time that son is going to be colorblind. And if we're looking at a daughter, in this case, 0%. And so that we see that with X-linked traits, males are significantly more likely to have be afflicted with these disorders and things because all they need is one recessive allele to express that trait. While females, in order for a female to be colorblind, she has to have 2 recessive alleles. And so when we talk about in lecture things like hemophilia, we see that it affects males far more often than females, and colorblindness. If you know people that are colorblind, any type of colorblind, you'll find that males have it significantly more often than females. Okay, we're going to look here at hemophilia in number 12. Okay, and so we've got a mother that's a carrier. And so as we do with all sex-linked Punnett squares, we have to put our female, we have to put our male. Mom is a carrier. This is hemophilia, so we'll use an H. Carrier means heterozygous. She's got the normal allele, so she doesn't present the disease, but she's got the recessive allele. She carries it. Okay? A father has the disease, so he's just got the one recessive allele. He's got the disease and nothing on the Y because this is an X-linked trait. Okay, so we're going to look what are the odds they have a child with a disease. What are the odds one of their sons has the disease? So we've got our X big H, X little h, X little h, X little h, X big H, Y, and then X little h, Y. So odds of a child with a disease. So how many here? We've got a dominant allele, no disease. Dominant allele, no disease. 2 recessive alleles, this daughter would have hemophilia. Recessive allele, but nothing to cover it up. This son has the disease. So 50% of their children would have hemophilia. And then what are the odds one of their sons? So specifically, if we're looking between these two sons, what are the odds? 50%. And so we have an interesting thing here that in order for a daughter to get hemophilia, the dad has to have that disease, okay, no matter what. And then the mom also needs to be a carrier. Otherwise, daughters will never get it, okay? A carrier, or the mom could have the disease as well. But dad has to have it for a daughter to have that disease. So those are our examples of inheritance. Doing these various Punnett squares. You will need to do this on your quizzes and potentially on an exam as well. So make sure that you're familiar with these things, practice them to get it down.